I would like you to take it away.
Absolutely. Hi, everybody. And you can all hear me? Thumbs up?
Yeah.
Great. My name is Brooke Berger. I'm a PhD student in ecology and in biology in Colleen Webb's lab. And I'm very pleased to be introducing our distinguished ecologist speaker, Dr. Ezenwa, today. I first met Dr. Ezenwa in 2018 when I was visiting the Odum School of Ecology as a prospective student. And she drove me and some of the other folks to dinner. And on that drive through Athens, I got to chat with a little bit about her research. And even on that short, little drive, I was able to see how fascinating her work was. So when Colleen suggested that I invite a distinguished ecologist speaker, someone whose work I want to learn more about, Vanessa instantly popped into my head. So this visit has been over a year in the making. She agreed to do this a long time ago, but with COVID, we had to postpone it for over a year. So we're very lucky to have her here today. So with that, let me tell you a bit more about our speaker. Vanessa received her BA in biology from Rice University and a PhD in ecology and evolutionary biology from Princeton. She's currently a professor at the University of Georgia and holds joint appointments in the Odum School of Ecology and the College of Veterinary Medicine. She's also the program director for the IDEAS program, which is a NSF-funded graduate student training program that focuses on studying infectious diseases at multiple scales, which is right in her wheelhouse, and provides training for students on quantitative methods and notably also science communication and a lot of professional development opportunities. Vanessa is a disease ecologist, and she gets to work both in the lab and the field and focuses on linking disease processes across biological scales, which includes how behavior and physiology at the level of the individual can scale up to influence populations and communities that are impacted by infectious diseases. And I believe this theme is reflected today in her talk. So we are very happy to have her here. And without further ado, I will turn it over to our speaker.
So thank you, Brooke, for that very kind introduction. And I'm glad that I could finally make it, if only virtually, to see you all today. Yeah, so I'm excited to have this visit in this form, at least. And I look forward to chatting with some people tomorrow as well. So today I'm going to focus on some work that my lab has been doing for a number of years, studying the sort of process of co-infection. And I think this work reflects somewhat some of the points that Brooke brought up, because our goals in this co-infection work is really to understand how things happening within individuals might influence processes that are happening at larger scales, in particular disease transmission at the population level. So with that, I will just get started. Let's see here. Okay. So co-infection or concurrent infection essentially is the norm rather than the exception in most organisms. So most organisms out there are infected with multiple parasite and pathogen species simultaneously. And as one example of that, I like to show this graph here, which is actually a plot of infectious diseases, particularly helminths, so worms and protozoans, pathogens like malaria, for instance, in a human population. And on the x-axis, it shows the number of different parasite species affecting individuals in this village by age class. So ranging from these lightest shaded boxes, so in children less than 5, all the way to adults over 40. And the y-axis shows sort of the prevalence, the proportion of the population that's infected with these various parasites. And one thing that jumps out on this graph immediately is that even at very young ages, so children ranging in the age from 5 to 9, so these lightest shaded bars, that over 90% of them are already infected with at least 2 or more parasite species. And this pattern only gets stronger as individuals age. So by the time people in this village are 40 or more, most of them are infected with 5 or more parasites. So more than half of individuals over 40 sampled at one point in time have 5 or more worm or protozoan species that they're infected with. And it's important to note that this is only a small fraction of all the parasites and pathogens that might be circulating in this population. So clearly, co-infection and sort of the simultaneous occurrence of multiple parasites and pathogens at once is the norm, even in human populations. So what this means really is that just like free living organisms we know coexist in communities made up of multiple species, that parasites and pathogens sort of experience the same thing. And since we know that interactions between different species and communities is key to driving ecological and evolutionary patterns, the same probably holds true for parasites and pathogens inhabiting a single individual host. But we don't really fully understand how these types of interactions inside the host environment work. And so there's a lot of unaddressed but really fascinating questions. And some of those are things like, what are the range of mechanisms that drive interactions between parasites? What are the things that cause these interactions? And also, what are their consequences? Also, we don't really know or have any sense yet, in general, what should predict which co-occurring parasites should interact most intensely. And finally, one really fascinating question that I'm particularly interested in is, how do these within scale interactions actually influence population level patterns? So can these interactions happening within a single host actually have some influence on the population level spread of parasites and pathogens? So studies in laboratory animal models suggest that helminths or worm parasites in particular can engage in particularly strong interactions, especially with microbial pathogens, so things like viruses and bacteria. So there's been a range of sort of emerging studies showing just how intensely helminths might be interacting with a range of different viruses, so both in animal and human populations, so things like HIV, etc. And so one reason that studying helminths in particular, why these strong potential interactions between worms and microbes might be quite relevant broadly, is that worms are actually extremely common. And so this graph here on the right is actually of the percentage of the world's human population infected with worms, right? So a third of the world population of humans are infected with helminths. And so this figure here shows the helminth ascaris, so a common roundworm infecting people, and nearly 1.5 billion people in the world are infected with this worm. And so worms are widespread both in human populations as well as in animal populations, be they wildlife or domesticated animals. So what this means is that there's ample opportunity for worms in particular to co-occur with a wide range of microbial pathogens. So another reason that worms are quite important is that they have profound effects on the immune system of mammals. So if we think of our immune system very simplistically as having two main arms, one arm that fights off intracellular microbes like viruses and some bacteria, and another arm that fights the extracellular things like these worms. We know that, right, these two arms are mutually inhibitory, such that when you're infected with chronic worm infections, they can downregulate your immune response to those microbial pathogens. So basically, they tip the scales in favor of the body's defense against worms and away from defense against microbes. And so what this means is that chronic worm infections have the potential to suppress our immune response to a wide range of microbial pathogens, potentially enhancing the susceptibility of individuals to these microbes, as well as the progression and severity of diseases. And in fact, in human populations, we've known for a while that co-infections with worms are linked to more severe infections of things like HIV and tuberculosis. So what's quite interesting is that the public health community, in the public health community, there have been sort of debates for a while about the implications of these interactions between worms and microbes for disease control. And one thought is that we can potentially use widespread and fairly cheap drugs that protect us against worm infections as an indirect means of controlling microbial pathogens. And so this idea of, HIV helminth co-infection, should we deworm, basically sums it all up. Maybe, right, we could indirectly target and improve outcomes of HIV in people, for instance, by intervening with anti-helminthic drugs, right, which are quite widespread, fairly cheap, and very effective. And so there have been calls for sort of using this type of indirect intervention with worm therapy, anti-worm therapy, to address the big three human diseases, which are HIV, tuberculosis, and malaria. So of course, worms also have direct negative effects on their hosts and on people. And in humans, in particular in children, worm infections have been associated with malnutrition, with stunted growth, et cetera. So there's a lot of impetus for, right, having large-scale treatment programs for worms. And in fact, and this is on the rise, right? Because as I said, there's effective and cheap anti-worm therapies that can be applied broadly. So drug companies, for example, like GlaxoSmithKline, actually donate 1 billion tablets per year to these mass drug administration programs, where the goal is the elimination or eradication, in some cases, of a range of helminthic infections on a global scale. Coupled with sort of these massive drug programs, right, there's large-scale education programs administered by the World Health Organization, and agencies like USAID also help in sort of the trying to massively get people out to administer these drug programs and potentially enhance the eradication and elimination of helminth infections worldwide. So worm management is not only an issue in humans, obviously, it is huge in animal populations. In animal populations, particularly domesticated animals, the widespread use of anti-helminthic drugs, so things some of you may have heard of, like ivermectin or fenbendazole, are used heavily in the agricultural industry, right? Because worms cause a high level of production loss, and a wide range of drugs are heavily used to counteract those production losses. Now, the problem is that more recently, high levels of drug use, anti-helminthic drug use in livestock populations has led to the emergence of drug-resistant parasites. And so alternative strategies like breeding animals for resistance have emerged. And in fact, these strategies have been quite successful in places like the Australian sheep industry, where breeding animals over time for low levels of worm egg shedding have been quite positive, and there's high heritability in this trait, right? So management for worms is not something unique to humans. Similar questions arise in animals. And so given what we know then about these potent immunological interactions that can happen between worms and microbes, one outstanding question that I'm particularly interested in is what then are the consequences of such widespread management and control for worms for microbial pathogens? In particular, what's the outcome in terms of the severity of disease for the host, and what does that mean for transmission dynamics of the microbe? It could in fact be a win-win situation where if we eliminate worms, we potentially enhance the immune response of the host. So we get rid of this immunosuppressive effect of worms, and that enhances host morbidity and mortality, and controls at a population scale microbial pathogen spread. But right, studies that connect what's happening within individual hosts with the immune response and these population level responses of pathogens to that interaction effect are rare or have been challenging to do. So that's sort of the background that has led to this line of research in my lab, where we're particularly interested in co-infections between gastrointestinal helminths, so GI worms, and bovine tuberculosis and we study this in wild African buffalo in South Africa. Wild African buffalo, I get this question a lot, might seem like an unusual system in which to try to study this question, but there's a lot of reasons that make it quite a good system to study this in. So interaction between worms and human tuberculosis, Mycobacterium tuberculosis, which is closely related to bovine tuberculosis, which is the pathogen we study, is well studied, right? The interactions between these two things in laboratory rodent models, in humans, and in cattle. So in a biomedical context, we know quite a bit about the interactions between these two pathogens. But wild buffalo provide this nice opportunity to expand this work and really understand the ecological and evolutionary context, how these interactions operate and what the consequences are on natural free-ranging populations, something that has been a gap because in laboratory models, we can't quite understand how, basically variation that's outside the lab influences this interaction and what the consequences of that are. So another reason why this system is quite interesting is that bovine TB, so Mycobacterium bovis, is an invasive and zoonotic disease in sub-Saharan Africa. So bovine TB was initially introduced into sub-Saharan Africa by cattle. It spilled over into wild buffalo, who are very close relatives of domesticated cattle. And buffalo now serve as the primary reservoir, right? So they maintain this pathogen in the system. It now spills back into livestock populations, so it threatens the agricultural industry in many countries in sub-Saharan Africa, and it also can infect people, right? So I said that M. bovis and M. tuberculosis, human TB, are very closely related. Well, they're so closely related that we can get M. bovis, and it's indistinguishable in terms of symptoms from M. tuberculosis. So it's thought that in many areas where people have close contact with their livestock, for instance, some important, significant fraction of TB cases in people might actually be due to M. bovis rather than M. tuberculosis. And so given sort of African buffalo's role in tuberculosis maintenance and spread in sub-Saharan Africa, TB management in buffalo is actually a primary concern in wildlife managers. So understanding how endemic parasites and pathogens that were in the system prior to the invasion of bovine TB influence the transmission dynamics of TB can only enhance our understanding of how to manage this disease in these populations. So today, my talk is going to focus on sort of two elements of worm management and controls: antihelmitic treatment, so treating animals with drugs to reduce their worm infections, and also host resistance. So this idea that there's some natural level of resistance in livestock, you might breed to enhance that resistance in a population, and trying to understand what are the consequences of this form of worm control on bovine TB. at both the individual host level as well as the population level. So starting with our work on antihelmintic treatment, so we approached this study using an experimental framework. And the key question here was to set up an experiment that might give us insight into whether an interaction between worms and TB, where if we could remove worms as has been predicted by lab studies, and this modifies the immune response of hosts to TB infection, whether this could actually impact survival of the host, susceptibility to infection, both of those things or neither of those things. And the goal was really simple, right? So we tracked 200 buffalo over a 4-year period. Some of them were treated with a long-acting antihelmintic drug, fenbendazole. And our goal was to understand what's the impact of treatment on infection risk, your probability of acquiring infection, and also your probability of dying once infected between the treated and control individuals. By understanding these two simple quantities, we might be able to understand what impact worm treatment has not only on individuals, but at the population level. And in fact, that impact might be quite different depending on which of these rates are impacted by treatment. And that's something that laboratory experiments on rodents have had a difficult time trying to assess. Right, robustly estimating infection risk in particular in the lab can be challenging because you need natural exposures in order to quantify that. So our work on this was based in Kruger National Park in South Africa, in the extreme south portion of the park. And so TB was introduced or first discovered in Kruger sort of in the 1940s. And so in the southern part of the park, prevalence of infection is quite high. So about 40% of all buffalo in the southern part of Kruger are infected with TB. So it's a highly prevalent pathogen. So we sort of situated our experiment in this high prevalence area. And so the basic experimental design for this study was set up like this. So we caught 100 animals who were designated as treated. 100 were designated as controls. I have to say all of these individuals at initial capture were tested for tuberculosis, right? If they had to be TB free to essentially be enrolled into our study. And all of them were female. This question comes up a lot. So we eliminated males from this study because male buffalo are highly aggressive, and it's really hard to track males over time. All of these animals had radio collars, right? So we could recapture them at fixed intervals. And male buffalo can actually get their collars off by doing a lot of aggressive things. So this study only focused on females. So treated animals were given a long-acting drug twice a year. And I actually brought a prop so people could see this. So this is what this bolus looks like, just like it is in the picture. So it's this cylinder with rings that you insert into the rumen, right? And it slowly, right, administers this drug over a period of about 4 months. So the nice thing about ruminants, you can put things in their rumen and it sits there. And so this is a treatment designed for cattle that works perfectly well in these buffalo. So we could essentially expand our recapture interval to 6 months or approximately 180 days. And control individuals got nothing. And so at every recapture time point, we measured a lot of things, but the key things we were interested in measuring were the number of worm eggs these individuals were shedding in their feces. So these GI worms are fecal oral transmitted parasites. So it means hosts ingest them with their food. The worms develop in the GI tract, they mate and lay eggs, and those eggs emerge in the feces of the animal, and that's how the cycle continues. So by assessing the number of eggs of these worms being shed in the feces, we can get a proxy of how infected the animal is. And this told us about the efficacy of our worm treatment. So we also measured the immune response. In particular, we were interested in this messenger molecule called interferon gamma, which is a marker of tuberculosis immunity, right? So our key objective, one of our key objectives here was to see whether worm treatment could actually enhance their anti-TB immune response by getting rid of that worm immunosuppression. And finally, at every capture, we tested them for TB, right? So since our primary concern was if and when these individuals acquire TB infection. So for some basics on the worm population in these Kruger buffaloes, so about in our first capture, about half of individuals, all individuals were shedding worm eggs, so actively infected with worms. The typical community of worms, right? They don't have just one worm either. They have a community of worms, was comprised of 7 different species, but dominated by 2 genera, this genera Cuperia and another one Homunculus. And these 2 worm parasites are well-known parasites of livestock, which sort of gave us really good clues about the life history of the worms themselves. And one thing that will arise later that's quite important is sort of this distribution of worm egg shedding in these hosts. And so, as I said, we measured the number of worms that are shed in feces as a proxy of the worm burden, the number of worms in the gut of these animals. And from a subset of animals, we verified that association, right? So we looked at whether worm egg counts were actually a good proxy of the number of adult worms, and in buffalo they are. So we know that higher worm egg shedding equates to a higher burden in your gut. But what we see here is this classical negative binomial distribution or aggregated distribution of worms in the population. So most individuals are infected with quite low burdens, and some have many. So this is common parasite distribution in any population, and this point will become important a little later in this story. So keep that in mind, that there's this aggregated distribution of worms. So one important thing is that we assessed over the time frame of our study the efficacy of our treatment, and our treatment was indeed effective at reducing egg shedding of these worms throughout the study period. And interestingly, our treatment regime was as effective as human mass drug administration programs. So we know that we did quite a good job at limiting the worm infection. So then our first question was, so if we can get rid of worms or at least minimize them effectively, well, what's the impact of removing the worms on the host immune response to a microbial pathogen, in our case, particularly TB? So to study this, what we did was that we challenged the immune system of these buffaloes, so in vitro in a tube, and measured the response to tuberculosis, basically the simulated response to tuberculosis, measured as the amount of this immune messenger molecule, interferon gamma, that they secreted. And so what we saw very clearly is that when we treat animals and give them that antihelmitic bolus, that we can enhance their anti-tuberculosis immune response. So this has been well documented in lab animal studies, that if you remove worms, you can enhance different components of the immune response. So it was not surprising in that regard, but it was the first demonstration in a wild population that, you know, we're seeing the same thing. But of course, we're all mammals. This is, I think, a very, you know, generalized response that we likely expect in most mammal species. But nevertheless, we showed that, you know, removing worms can have an effect on this opposing immune response to microbes. So given this, the question then was, okay, we can effectively enhance their anti-tuberculosis immunity. Does this actually have a protective effect when it comes to protecting them against tuberculosis? And surprisingly to us, the answer was no. There's no effect of treatment, or absolutely no effect of treatment on TB infection risk in this population. To orient you to this survival curve here, so on the x-axis is time in days from the beginning to the end of the study, and the y is the proportion of proportion of our study animals converting to TB positive. Controls are in blue and treated are in red. And as you can see from these interdigitated lines, there's absolutely no effect of treatment on an individual's probability of getting infected. And in fact, the hazard ratio is 0.98, so close to 1. So the odds of getting TB, if you're treated or controlled, are essentially 1 to 1. There's absolutely no difference, despite the fact that we could enhance their anti-tuberculosis immune response. So the next question was really, okay, if we can't affect their response to TB infection, right? We can't prevent them from getting TB. What happens once they have TB? And it was here that we saw a drastic effect of the treatment. And so again, this graph on the Y is showing the proportion of individuals surviving after TB infection. So this graph is only those who got TB. And the time in days here is now from the onset of your TB infection till death or the end of the study. Again, blue is control and red are treated individuals. And we see this huge gap here. And so essentially control individuals were 9 times more likely to die of their TB infection. And this is really a massive effect of just removing worms. So it turns out that by treating you for worms, we enhance your immune response. That immune response does not protect you from getting TB, but once you have TB, there's a huge impact on your survival. So having the TB and not having to battle your worms at the same time is a huge positive impact. And so we were then interested in this question of, okay, so we clearly have seen that there's individual level effects of worms and removing worms on TB, but what are the population level consequences of this antihelmitic treatment then? So to do this, we focus on calculating what's called the basic reproductive number, or basically the fitness of TB. And R0, this quantity here is defined as the number of secondary cases that an average infected individual causes over the time course of their infection. So till they die or until they recover. And essentially R0 tells us about the capacity of a population to spread in a pathogen to spread in a population. And this figure just shows the R0s for a range of common human pathogens. So we can see, for instance, that Ebola has an R0 of 2, which means that the average Ebola patient infects 2 others before they die, where measles has an R0 of, there's something in the chat. And measles has an R0 of 18, where an average one measles patient, right, can infect 18 others. And so measles clearly has a capacity to spread much faster and further than does Ebola. And just for some, interest, this is SARS coronavirus 2, right? So the causative agent of COVID-19. So the original virus, not one of these variants. And the R0 recently estimated for SARS-CoV-2 is between 2 and 4. So just to calibrate that. So what we were interested in with estimating R0 is basically, right, to understand the population level spread of a subpopulation of buffalo who are treated compared to those who aren't, to understand, well, how do those individual level effects we quantified in the experiment scale up to population level spread of TB. And so we can use a simple susceptible, infected, recovered framework to sort of visualize how we estimated R0 in our population. And so individuals in a population, when we think about diseases, can just simply be put into compartments. So susceptible individuals who are not yet infected but can get infected, infected individuals, and then recovered individuals. So you will see that recovery is grayed out here because tuberculosis is a chronic infection, and without treatment, you can't recover. So clearly no one's treating these buffaloes, so we grayed out recovery. And so susceptible individuals enter the infected class, through at some rates that we can quantify as beta, the transmission rate. They leave that class in two ways. They can die a natural death, so mu here, or they can die due to the disease alpha. And so we can estimate R0 in this case as simply the ratio of new individuals who become infected. So the rate at which new infections arise, beta, divided by the infectious period, which is the sum of the rates at which they leave the infected class. And so in our situation, just qualitatively, we know that from the experiment, that worm treatment did not affect the numerator beta, but it actually reduced disease-induced mortality. So it reduced the denominator. So interestingly, this suggests qualitatively that treatment has the capacity to enhance R0, which is counterintuitive to what I would say sort of general consensus was about how these within individual effects might scale up. So when we went on to estimate R0 in our control and treated subpopulations, we found that for control individuals, R0 was around 2. So an average buffalo infects 2 other buffalo with TB before they die of the disease. In contrast, we actually estimated that the R0 for treated individuals was more like 15. So this is a massive increase in R0. And now you're getting a single infected buffalo infecting 15 others before it dies. And this unanticipated outcome is solely due to the asymmetrical effects that worm control is having on TB infection risk. So your probability of getting TB in the first place, and then your mortality. So unfortunately, TB doesn't slow down, worm treatment doesn't slow down TB acquisition. You're still as likely to get TB. But then it enhances your survival. So whereas that's a good thing at the individual level, actually at the population level, that's a cost because it enhances, right, just the infectious period and allows these buffalo to hang around longer, infecting others. And so studies in lab animal populations are not able to measure this natural transmission risk. So for a long time, I think it was assumed that the impacts of worm treatment would be symmetrical, right? So if worm treatment was reducing or influencing infection risk, that you'd have a symmetrical response on mortality. But it turns out that those effects are totally different, right? In our case, there's no effect on one and there's a massive effect on the other. And so I think this result highlights sort of a public health conundrum, which is that we might control worm infections, right? Controlling worm infections enhances individual level morbidity and mortality. So that's a good thing. At an individual level, to use worm control is this indirect way to intervene with more potent microbial pathogens, in this case, tuberculosis, but it does come with a population level cost, which is that the pathogen might have a better opportunity to spread at the population level. So it's one thing we need to consider when we sort of, if you think back to that slide where people are proposing using antihelminthic drugs as a means to control microbes, these asymmetrical effects might be a thing to consider, that it's not a win-win situation at all levels of organization. Okay, so with that, I'm going to turn to sort of the second arm of this work, which was looking at the role of natural host resistance and how that might influence tuberculosis. Um, and our work focused on this topic really came about due to just observing some interesting patterns we saw in our field data with respect to how antihelmintic treatment and natural worm burdens influence anti-tuberculosis immunity. So I'm going to walk through these patterns, some of which you've seen before, just to set up these interesting contrasts. So if we think about antihelmintic treatment, I showed before that when we treat buffalo for their worms in Kruger, we saw this enhanced interferon gamma response. So we saw enhanced anti-TB immunity. And this was actually the second time we've seen this. So previously, we did a small scale study in another buffalo population in South Africa, in Shushului and Folozi Park, where we saw the same thing. So in this graph here, uh, we treated animals once over a 6-month period. So we go from pre-treatment to post-treatment. Everyone, both treated and controls, increases you will see because we went from the dry season to the rainy season. So basically an increase in their nutritional resources caused everyone to improve immunity slightly, but still treated individuals improved much more than untreated individuals. So again, this is a very general result. When we apply worm treatment, we see enhanced anti-tuberculosis immunity. So what's interesting is that when we look at the patterns of just their natural worm burden, so how many worms these individuals have naturally, in that Shushului population was when we first noticed that the number of worms you have is actually positively correlated with how much interferon gamma you produce. And in Kruger, we see the same pattern. So there's a positive correlation between these things, which suggests that when you have a high worm burden, you have a high level of interferon gamma production, and a low worm burden is associated with low interferon gamma production. So that was quite confusing to me anyway at the beginning, where I think, okay, well, when we're reducing your worms with drugs, we're boosting your interferon gamma response. But when we go out and just look at correlations between worms and interferon gamma, it's the opposite direction. And so if we go back to this aggregated distribution that I talked about, where a lot of individuals have these relatively low burdens and some individuals have these high worm burdens, we actually find that this, so since we measured individuals over 4 years, repeated measurements, that these are highly repeatable measures, right? So if you're a high shedding buffalo, you're a high shedding buffalo. If you're a low shedding buffalo, you're a low shedding buffalo. And of course, environmental factors and all these things play a role. But even if we account for all of that, there's repeatability in the shedding pattern. So this suggests that there's these interesting worm phenotypes emerging, sort of high phenotypes, so individuals who have lots of worms and individuals who have fewer worms. And so we use sort of a statistical approach to look for natural breaks in this data set, and found that, you know, there's a natural break at about 100 eggs per gram shedding, and about 60% of individuals fall into this low class and 40% into the high class. And what's interesting about this, in this phenotype, the high and low is also repeatable. And so what's interesting about this is this phenotype is actually associated with their anti-worm immune response in their gastrointestinal tract. So we were able to, at the end of this study, look in vivo at a subset of our animals and measure their response to worm infections at the site of infection in the gut. And this figure here is just showing various immune molecules and proteins that help defend us, mammals, against worms here. And so we looked at one key cell, these eosinophils, which are really important in worm defense as well as antibodies. And here you see that when we look at high and low phenotype animals who maintain low worm burdens have significantly higher numbers of eosinophils in their intestine and stomachs. And likewise for immunoglobulin A, which is an important antibody that defends against worms and enhances worm control. So these worm phenotypes are actually associated with the level of resistance that these animals naturally have to their worm infection. And we now have some preliminary data that suggests that these worm phenotypes potentially have a genetic basis. So we particularly focused on one candidate gene, BL4, which is close to the interferon gamma gene. And that's because in a lot of domesticated animals, particularly sheep, as well as free-ranging soy sheep, polymorphism at this IFNG gene has been linked to variation in worm resistance. So we decided to start with this particular gene region for that reason. And of course, IFNG, this gene, is the gene that codes for this protein that we're very interested in as well, which is the protein that helps in immune defense against tuberculosis. And so at BL4, we had previously found in our HIP population that polymorphism basically at this allele 169 was associated with variation in your worm burden. And likewise, we see that in the Kruger population as well. So these populations are totally distinct. There's no gene flow between these populations, and we see two other alleles, one of them 141 in this population, that are linked to variation in worm burden. And so animals who have allele 141 present tend to have lower worm burdens, and low worm burdens, and allele 141 is sort of overrepresented in individuals that are in the low resistance class compared to individuals that are in the high non-resistant class. So again, this is just one gene region, and we're interested in sort of expanding our work on this issue of the genetic basis of worm resistance in this population, but it is suggesting to us that this phenotype that we see very clearly, that's repeatable, has a genetic basis. So for this reason, we are actually interested in understanding what role this phenotype, like how this phenotype translates to TB infection risk and mortality. So doing exactly what we did with the experimental treatment, we simply looked at what the consequences of being a low FEC animal, so a low worm burden animal. You could also think about the low as the resistant animals versus the non-resistant animal. What was the impact on probability of infection and mortality? And of course, we controlled for the presence of the antihelmitic treatment. So like the antihelmitic treatment, we actually found that worm phenotype had no effect on TB infection risk. But resistant hosts were more likely to die when infected with TB. So again, this is the second time that we're seeing that actually nothing we can do by manipulating worms is impacting the host's likelihood of getting infected. And that might have to do a lot with the social organization of these animals and the exposure risk to TB. So nothing we're doing by manipulating their immune response is impacting susceptibility. And most of the impacts we see are on severity of infection once you get it. And in this case, we see that, again, to orient you to these graphs, this is time in days from the onset of infection till death or the end of the study, and the proportion who die. Significantly more resistant or low phenotype individuals die, and in fact, they die at a rate that's 5 times higher than non-resistant individuals. So what is fascinating here is that when we treated individuals for worms and artificially reduced their worm burden, we enhanced their survival for TB. When they control the worms themselves, their survival tanks, right? So low worms alone can explain this, because controlling worms by yourself is actually a cost when it comes to TB mortality. But if we artificially control their worms, they reap that benefit. And so one interesting thing is that the effect of worm resistance and treatment are actually independent and additive, and that's quite interesting. So this graph is really busy, but I'll walk you through it. So time here on the x-axis is again from in onset of TB infection till death at the end of the study, and this is the proportion surviving. Treated individuals are in red, controls are in blue. The solid lines are the high individuals, so that's the non-resistant phenotype. The dashed lines are the low individuals or the resistant phenotype. And so one thing that we can see clearly is that treated individuals overall do better than controls, right, in terms of mortality risk. But strikingly, even among treated individuals, we're seeing a gap in mortality. So in parentheses, here's the cumulative mortality in that group over the course of the study. And we see that low individuals, so these naturally resistant individuals to worms, experience 14% mortality, whereas these treated individuals who are non-resistant, none of them died throughout the course of the study. So this actually suggests that this low resistance phenotype, and so again, this is resistance to worms, comes with a cost even if you don't have any worms, right? Because we treated them. So even though they're not experiencing an active sort of ecological interaction with those worms, this genetic this genotype they have, or like this genetic effect of being resistant to worms, still comes at a cost when you get infected with tuberculosis. And so the additive negative effect of worm resistance and a lack of treatment is best seen by comparing these treated high individuals who have the drug and also are non-resistant to worms. None of them died over the course of the study, which, good for them. And then these control low individuals. So these are untreated individuals who are resistant to worms. So nearly half of them died when they got TB over the course of the study. So this is the class of individuals who experience the worst cost of TB. And interestingly, right, control individuals, that's the natural state of buffalo. And if you think back to that aggregated distribution I showed you, 60% of buffalo out there in the low class, right? Most buffalo in this natural population are actually resistant to worms because they are, they have co-evolved with their worms, right? Worms are in the endemic infection in this case, and TB is actually very new. So the sort of baseline state of buffalo is to potentially be very vulnerable to TB infections for two reasons. One, because you're actively infected with worms, and two, because of your evolved resistance to worms. And so to sort of summarize this, so what we see here is that natural resistance enhances mortality of TB infected individuals while treated while treatment reduces that mortality. And we're really interested in now in understanding why this happens. So we've done some work to gain some clues about this, and I'll summarize that sort of as the last step here. And first, I will give a little primer of TB progression for those who don't know that much about tuberculosis. So this figure obviously is human tuberculosis, M tuberculosis, but bovine TB acts in the same way. So TB is a classical respiratory pathogen, which you can inhale or, you get infected through the nose or the mouth. The lungs are the primary site of infection. And so the TB bacteria basically get into the lungs where they replicate in cells there. And the primary immune response to TB is to create these lesions, also called granulomas. And essentially what these are immune cells that encapsulate that bacteria, and they inhibit the growth of that bacteria. So they basically, freeze the bacteria so they aren't replicating. And so that's how our bodies control TB in the lung. But for many reasons, right, we can lose control in the lung, right? So our immune responses might be suppressed in some way, whatever reason. And these lesions essentially open up or they burst, and that allows the TB, right, to get out of that lesion and continue causing new infections throughout the lungs. And right, at some point, the TB can also, if not well controlled in the lung, it can circulate throughout the rest of your body, including to the lymph nodes. And that's when you get what's called disseminated TB. And that's actually what causes TB mortality, is that TB has now spread from that primary site of infection where it's under some control to other parts of the body. And it causes sort of wasting disease, and that's why TB in the past had been called consumption, basically, right? It's consuming your body. A lot of organisms can get TB in the gastrointestinal tract, in a lot of the respiratory system. So disseminated disease ultimately is what causes the mortality. And so we can essentially assess, right, how TB is progressing in an animal by counting up these lesions in the primary site, the lungs, and also looking for lesions in disseminated sites like the lymph nodes. We can see how big these granulomas or lesions are. We can measure what immune cells are inside these granulomas, all to understand what stage of the disease you're in, how advanced it is, and sort of how much morbidity it's causing. And so we did this with a subset of our animals at the end of the study to try to understand, okay, we know all of this stuff about the immune response in vitro, but what we don't know, what was missing was how are these animals actually responding in vivo to their TB infections when they were treated, or whether they're resistant or naturally resistant or not. And so just to show some of those results, so in the lungs, we focused on the lungs, the primary site of infection, and also the lymph nodes. So in the lungs, we saw that those low FEC phenotype animals, the resistant ones, tended to have more lesions in the lungs. So they tended to have more of these lesions, suggesting that there was more bacteria, right, they were losing control of the lesions, and the bacteria were replicating and spreading around the lungs. They also had more of what are called these multinucleated giant cells present in the lungs. And this shows, right, again, this fact that there's more granulomas spreading around the lung, and the animals have lost control somewhat of their TB infection at the primary site. And ultimately, these animals were in significantly more advanced stages of disease. So that's if you were a resistant animal. So if you naturally control your worms, for some reason, you could not control your TB in the lung. And surprisingly, we found that treatment actually had no effect on TB progression in the lungs. So the lungs of treated and controlled buffalo looked the same. With respect to progression. So treatment was not impacting anything in the primary site. When we looked at the lymph nodes, so we looked at multiple respiratory lymph nodes, focusing on one in particular, the retropharyngeal lymph node. We see that this is where treatment is actually having a strong effect on the disseminated disease, right, at these other sites of infection. So control animals had significantly more positive lymph nodes. So this means more of their respiratory lymph nodes were getting infected with TB. And so they had more dissemination of the disease. Likewise, they again had these signs of lesions in the lymph nodes, these high levels of multinucleated giant cells, suggesting that they're in more advanced stages of disseminated disease, right? They lack control in terms of controlling TB at these other locations of infection. And surprisingly, worm phenotype actually had no effect on TB progression in the lymph nodes. So ultimately, what this suggests to us is that treatment effects. So when we give you a treatment to reduce your worms, the effects are manifesting in the lymph nodes. So it's something about the dissemination of disease. Natural resistance effects are actually manifesting in the lungs, so it's something about the primary site of infection. And so a little summary here. So what do we know so far in the system? So we know that both having worms and resisting them have consequences for host responses to microbial infections. And I think this is what's slightly new here, is that the old way we thought about worm TB and worm microbe interactions was that it's about an interaction between two things that are contemporaneously present. But this suggests that, the worms don't have to be there for you to also have this interaction. We know that antihelmitic treatment improves TB survival at the individual level, and this is because of some effect on treatment on disseminated disease, right? Treatment is helping to control the dissemination of TB, and that's how we see this mortality reduction. But, right, this nice individual level effect potentially comes with an unintended consequence, because the host lives longer, at least in the case of buffalo, and that means that TB can spread faster at the population level. So we also know that natural resistance negatively affects the host response to TB. And in this case, what natural resistance is doing is that it's suppressing control of TB at the primary site of infection in the lung. So something about being good at controlling your worms means that you have poor control of tuberculosis in the lung. And what's interesting here is that I think that this could have some profound implications for livestock in particular, where breeding for resistance to worms is an expanding practice because of the spread of drug-resistant worms. And so if this result is general, it actually means that breeding for resistance might make some livestock populations at least more vulnerable to some microbial pathogens, at least bovine tuberculosis. And then finally, we know that there's different pathways to low worm burdens, artificial treatment, and natural resistance, and these are associated with distinct immunological responses to TB infection. And this is something we just need to understand better, is like how exactly do these distinct immune responses work? So our in vivo work tells us that these things are happening in different tissues very differently, that the effects are manifesting, but we don't fully understand the cellular and molecular mechanisms that lead to this. So our ongoing work in this system there's a lot of questions obviously left to answer. So our ongoing work in this system is going to start what COVID allowing, I guess I should say, to try to understand what the mechanisms are actually that link natural resistance to TB severity in the lung. And so one of our hypotheses is that this has to do with key immune cell macrophages that are important components of the granuloma, and the fact that the worm-resistant animals might have a bias in what types of macrophages they have in their lungs. We're also really interested in in this idea that TB selection that there might be TB-mediated selection on worm resistance. So everything I've talked about here has been like one direction of how worms affect TB. But one really interesting thing in this system is the TB is invasive. There's high levels of worm resistance in this population. Well, what does the invasion of TB mean for selection on worm resistance? Nicely, Kruger is the perfect, I think, natural experiment for this, because TB arrived in the 1960s and took about 40 years to spread from extreme south to extreme north. So it gives us this space for time sort of substitution gradient that we can use to understand whether TB and the invasion of TB has had an effect on resistance in this buffalo population, right? So prevalence of TB is about 40% in the south and less than 10% in the north. So we're excited about trying to understand this reciprocal effect of TB potentially on worms. And then finally, we're also interested in the idea that there might be consequences here for TB virulence evolution as well. So the reason here is that TB might actually be affected by the fact that it's persisting in two types of individuals, one that's very permissive for TB replication, and those are the non-worm resistant hosts, and one that are not very permissive and die very quickly. So that means there might be strong selection on TB virulence because TB can replicate much faster and potentially be more virulent in those hosts, non-resistant hosts who are fairly tolerant of mortality at least. So our goal is to use this system and this natural experiment in Kruger to address these coupled ecological and evolutionary interactions between worms and TB that hopefully bring together what has been quite a disparate conversation about co-infection, where people are actually primarily interested in ecological interactions or primarily interested in potential evolutionary interactions. And this system is one where probably like most systems, where both things are happening in tandem, but we might actually have the opportunity to start understanding how these combined processes work. Okay, so with that, I need to thank, all of my collaborators on this work, who this work would not be possible without them. So Anna Jallis from Oregon State, Carrie Sakamoto, who's a veterinary pathologist here at UGA, Gordon Lukart, who's the population geneticist at the University of Montana, Sarah Budashak and Mauricio Seguel, who were members of my lab, who are now run their own labs, and Peter Buss, who is the Director of Veterinary Wildlife Services in Kruger, and South African National Parks more generally, who obviously without their permission to do this work in Kruger and without massive logistical support, we couldn't do this work. And also I'd like to thank a wide range of students, postdocs, both field and lab technicians and staff who all contributed to this project. And thank you guys for paying attention to this, maybe, endless Zoom talks, right? So yeah, thanks.
If y'all could unmute and give Dr. Ezenwa a hand, that would be great. That was so fantastic, so fascinating. I would love to take we would love to have some questions.
There were some things that arose in the chat when I was talking.
Yeah.
Maybe some of them were questions. So would you like me, there is some questions there. Should I start with that or wait for?
Sure, that sounds good.
So there's a question from Ashley that was about females being of reproductive age and how gestation affects their immune response. So that's a great question. And yeah, so we caught individuals sort of between the ages of 2 and 3 to maximize the likelihood that they would get TB during the time frame of our study. So many of our individuals transition to reproductive age. So we kept track of their reproductive status, and that was accounted for in all of our analyses. But indeed, reproductive status does influence your immune response.
Teresa Laverty, looks like you have your hand up.
Hi, Vanessa. Good to see you. And awesome talk. I was just wondering, I know from your research in Kenya that you were also kind of looking males versus females. And you said you were unable to look at males in this case for very valid reasons. But I was wondering if you would expect any sex differences there among worm infections or TB infections.
Yeah, so that's a great question. So yes, so in my other life, I do actually study some differences between males and females in ungulate populations. And one thing we know is that in many cases, in a lot of worm infections, there's actually male biased infections where males actually have more intense infections. In our HIP study, we did have males, and we did see that males sort of on average have higher infections, but we saw the same immunological effects in the males and females. So when we treat them for worms, that population was mixed sex, we see the same effects. So sex had no impact on this immune effect. And I actually don't think it would have an impact on any of the other results that we saw, because there's no, I guess, given that that immune effect exists, I think that it would only enhance the outcomes, right? That more intense worm infections actually mean potentially more intense interactions with TB. But of course, we haven't looked at that, but I have no reason to think that there would be a difference.
Great, thank you.
Anybody else at the moment? I have a question. So it looks like TB has spread pretty thoroughly throughout this range. And like, I'm wondering like what the infection rates overall are, you know, like is an animal invariably going to get it?
Yeah, no, so I mean, it also matters where you are. So where our study was based, there's a 40% prevalence. So, not all animals will get it ultimately, maybe slightly under half of them or something like that. So many animals I would say in our study population over our time frame, about 30-something% of animals got infected.
Fascinating that it's not higher. And then that leads into my follow-up question, which is like, say you could actually treat them all for worms. Would that be like, so there's the individual on the individuals wouldn't die as much, so which has a populational level effect if you're looking at just like overall population size. So if you're managing for population size, would it be good to treat them for the worms, or is it truly best to just like hands off, let that play out?
Well, I guess if you could treat them all for worms, so if you could treat them all for worms, they would still have, what I would assume is that TB could spread much faster.
Right.
But you keep them alive longer. Yeah. I guess it depends on what you're managing for. That's what I think, right? So like, what is your ultimate goal? Is it to have infection-free animals, or are you okay with having TB-infected animals who are still, right? They're still alive. So I guess those are all of the questions that I think that make this complicated. It's really about what do you want to maximize. So do you want to maximize population size, or do you want to maximize, healthy animals? Or
thank you so much,
if
Jennifer.
that answers that. It's not
it's a hypothetical because it's not feasible to treat them, of course. Jennifer?
Thanks. Yeah, no, that was a really fascinating talk. Thank you so much for being here and sharing that. I actually had a couple questions, but I'll just pick one and stick with that. And it's kind of a follow-up to thinking about the transmission of worms from the buffalo to predators, for example, and what the rate of that is, if that even is a thing, and how that might be changed by treating the buffalo for worms. Does that make sense?
Yeah. So it does make sense. So the worms we treated for are worms of buffalo and other ungulates, right? Okay. So they're not worms that their predators can get. There are in fact worms that are trophically transmitted, which means that the main host is a predator, and the predator has to eat the buffalo to get those. So we didn't focus on those type of worms in our study.
Okay. And then if you don't mind, I do have one other question. And that is thinking about, that correlation between the worms, which they've been, co-evolving with for so long, versus TB, which is so new. and I know it wasn't part of your talk, but have you looked at any other pathogens that maybe have been around for a while? And have you thought about how those might be shifting at all with the presence or absence of worms?
Yes, so that's a good question. So yes, we did indeed measure some other microbial pathogens, and we never see effects as strong as with TB. So I do think that this this interaction between something that has been around a long time, these worms, and something new, TB in the system, may be playing a role in this. But on the other hand, we also know that, people have seen in laboratory systems anyway, and there's some cross-sectional evidence in people of interactions between worms and tuberculosis, human TB. So there's certainly microbes for which we see these effects more strongly than others. And of the viruses and bacteria we've looked at in our system where we see these strong effects, TB is the only one that's really been studied in any other context. So, so I'm not surprised we see these TB effects, but I don't know what to make of the fact that we don't see them as strongly for some other things. Now, whether that's something about the particular microbes for which these interactions are most important. So I raised that question at the beginning, like when do we think these interactions should be most intense, right? So is this something about the pathogens, or is it something about how long they've coexisted? So those are the types of questions that we hope to be able to continue to try to understand.
Yeah, that's going to be exciting. Thank you.
We've gone.
Looks like there was a question in the chat. Someone was wondering what these TB infected individuals were dying from, whether it was predation or hunting if they left the park, or if it was actually from TB.
Okay, this is a great question that always comes up. So the proximate cause, I guess, of their mortality is predation, 100%, right? Because almost no buffalo just drops dead. They're all killed by lions. But there's a huge, most of the animals the buffalo that die, that are killed by lions, have TB, right? And they weren't treated. So I guess that raises the question, right? That that's another thing that I think is quite interesting in these systems is that the whole community context is actually important to see these outcomes. If you just have buffalo who have no pressures, right? There's like no predators, they might have lived for double the time, right, before they died. And so, yeah, so that's an excellent question. And ultimately, it's lions that get buffalo. They don't just drop dead because, right, the time course of a chronic TB infection, for it to impact the individual so much that you just die, that would take years.